great to be back. I hope you guys have been having a good workshop or course so far. And again, please, if you have any questions during the talk, just feel free to interrupt me, and I'm happy to explain a bit more about all of this. So for today, I wanted to focus on how to do, well, spatial single cell analysis in Python. And I will be focusing on our Squidpy software package that we've released, well, roughly like 1.5 years ago now. And this is supposed to be kind of an extension of ScanPy. So it works in the same ecosystem. It uses these undata data matrices and all of this, but it's extended essentially to hold all the spatial information. And on top of this, we then provide a couple of tools to analyze the data. And as far as I understand, you've already used Squidpy in some of the previous labs, so you might already be a bit familiar with it, but now this would be the chance to really get into the details of what this package is providing. Squidpy is very much a joint work together with Giovanni, Palla, and Michael Klein, who are one of the leading developers in this project, and many other members from the TIES lab. So we're really, this was really very much like a joint hackathon project. This is how it all started. And now we're quite happy that this has already been taken up a bit by the community. And I'll also say something about how we plan to extend the software in the end of the talk. Okay, yeah, so this again is a short overview of kind of the Python ecosystem that we have for single cell analysis. And I already talked about this yesterday a bit, but essentially we have, this is all also in now the new, in this new organization where we try to pull together all the Python tools for single cell analysis. And we have ScanPy and Undata, which is for basic analysis, but then there's also other tools that are more focused on specific questions. So for example, we have the single cell velocity, the SCVelo for the trajectories. Then there's CellRank, which is like for probabilistic cell fate mapping. There's also SCVI tools, which is for kind of data integration using deep learning methods, and also SIRPy, which is for the immune cell receptor data. And what it was kind of missing in this whole ecosystem was something that could deal with spatial data. And this is where, and spatial data is actually something that is coming up a lot in the last years. I mean, it's now we're all kind of aware of it, and this is also why we're doing this course. But this is we are one of the really new methods that allow us to gain more information about about how cell systems work, how tissues work, and all of this. So for this reason, we developed this Squidpy software package. And essentially we're trying to really integrate this into the ecosystem by, like I said, using the Undata formats and keeping the same principles of modularity and scalability that we also have in ScanPy. So we tried also to name the functions quite similar of how you would use them in ScanPy, and also try to make it in a way that for you as users, it would be quite seamless. If you're kind of used to how ScanPy works, then Squidpy would work in a very similar way. Okay. So as you are all probably aware of by now, because in this course, it's also really nicely represented that spatial data is very diverse. So there is many different ways of how you could acquire the data, and you've learned all of this on Monday already. There is kind of these image-based technologies, or contrasted to the sequencing-based technologies. And there's also different, many different resolutions that you could acquire the data at, and also resolutions in the spatial sense, but also resolutions in the sense of how many genes or how many proteins are you measuring. And yeah, just to name a couple, there is Visium, for example. which it does kind of an in situ capture of the RNA molecules. Then there is SECFISH, which could only capture a few genes, and then now there's these extensions of MERFISH, for example, that allow you to really target many more genes, but it's still like a targeted approach. And then there's all these imaging-based approaches like IMC or CISIV, or also 4I that allows you to measure proteins. And there are quite a couple of tools that that deal with the pre-processing steps of the data, and I'm just listing a couple of those here. And what is important to note is that for Squidpy, what we aim to do is we aim to be downstream of all of these pre-processing tools. That means we don't really want to deal with acquiring, so with kind of processing the images in a way, with dealing with illumination correction and all of this, but really we want to start at a common, at the theme that all of these technologies have in common. That is that you essentially have a matrix of observations, of observations that could be cells, that could be subcellular observations by genes, or more generally speaking, features. And also you have a spatial relationship between all of these observations. This is kind of the common theme that we want to start from in Squidpy. So like I said, all these pre-processing tools would be kind of happening before this step, before this unified representation of the spatial data. So for this representation, like I already said, what we have is a spatial graph. So this is something that relates the individual observations that you have to each other. And we also provide something that's called the image container, that is essentially a representation for the high resolution tissue images that you may also obtain together with your spatial data. And this actually gives you the added advantage because of adding some more information to your analysis. So for example, you could use these tissue images to segment the cells, and then if you're looking at spatial transcriptomics, where you have these spots and there's many cells per spot, or well, a couple of cells, maybe 10 cells per spot, and you could actually segment and count the cells that you have per spot by leveraging the H&E stained tissue image. And this is something you could do in this Squidpy framework. And of course, having a real, knowing the actual number of cells that were imaged in a particular spatial transcriptomic spot, this is quite important for a lot of deconvolution methods, for example, or in general for downstream processing, to just know how many cells are you actually looking at in your small spot. Okay. So for and for this, so for the data representation that I just talked about here, we have one lab coming up. This is the spatial A-data lab. So this is this is something you can then kind of really get your hands on, and we'll provide a couple of examples of how to create these representations in the code and how to work with them, essentially, so you can also use your own data to and put it in there as well. Right. So downstream of these representations, once we have our spatial data in either a graph or an image container, there is a lot of different analysis tools that we provide with Squidpy, and we continuously aim to kind of be able to extend this when more and more tools are now coming up, because like with spatial data in general, the challenge, but also the nice thing is that it's a very open field, and there is not a lot of established best practice methods at the moment that that are used for this analysis. So when we wrote this toolbox, we just looked at what do people in general use and try to think of more tools from, say, computer vision or other related disciplines to try and make a nice comprehensive toolbox of things that you can use to analyze the data. So we have several methods that can quantify spatial variation of the tissue. So this is both for continuous covariates like gene expression, and also for discrete annotations, like if you have healthy versus diseased regions in the tissue or different cell clusters or something like this. So kind of methods that can quantify the spatial variation in this way. In addition, for the images, we provide a couple of tools for some pre-processing tasks like cell segmentation or and also visualization of the tissue image. So we have this interactive visualization plugin that is, this is a Napari plugin that you can use to visualize the tissue images. And there's also a couple of static plotting functions, similar to the plotting functions you get with ScanPy. Okay, so in terms of the graph, I'm now going to go a bit more into detail about the individual components, that is the spatial graph representation and things you can do with it, and the image representation and things you can do with it. So we provide a couple of different methods of how you can build the spatial graph. Because in general, you can imagine many different ways of building a graph, and here I'm showing a couple. So you can you can build a really regular graph just based on a square or a hexagon, or you can build more generic graph building methods where you just have a point cloud and try to connect points that are close to each other, using either a radius-based thing or the Delaunay triangulation, or you could use something like a KNN graph with a sufficiently low number of neighbors that you want to connect. And so for example, for the spatial transcriptomics, obviously a square-based, or depending on the type of assay, maybe also a hexagonal-based graph is the most adequate, because this is essentially how the data is structured. So it makes sense to represent the spatial relationships also in this way. But and there is also stereosig and Visium that fall in kind of the similar category, and Visium being the one that is hexagonal here. But there is kind of this imaging-based molecular data, which is like the Mibitov and the Murfish and the Sackfish, that where essentially each of the nodes of the graphs will be individual cells or pixels of this image, and then the neighbors can be determined by kind of the radius-based or more generic graph building methods. And this whole graph-based spatial context, sorry, one more animation here. This whole graph-based spatial context was actually used quite recently to research kind of cell-cell communication by describing the gene expression of a cell as a function of its neighborhood. And this is this enzyme model that, and I think Anna Shah will be here this afternoon to also talk more about the cell-cell communication and how you could do this on your own data as well. Right. So within Squidpy, there is a couple of different tools that we provide to extract spatial patterns in the tissue. So on the one hand, there could be continuous features, like, for example, spatial variable genes. So for this, spatial variable genes are essentially genes that are, well, varying across the space. And there is a couple of simpler tools to calculate it. So this is the Moran's I, for example, or the CPAL score. And this here on the slide, you can see an example of two spatial variable genes that are detected by, I think, the Moran's I statistic. And you can actually see that these are really genes that are expressed in one particular spatial region here. And in the labs, we will see how to do that with Squidpy. It's actually just one line of code to get these genes. Another thing you can do for continuous features is, for example, to check the ligand receptor interactions. So this is something that is maybe interesting if you want to figure out what pathways are activated in your tissue and where it varies across the tissues. And for this, we re-implemented the Cell4DB score for this ligand receptor interaction test. So this is done by just querying the OmniPath database for known ligand and receptor interactions and calculating a permutation test. And essentially you can use, for this, since you have the spatial information available, you can use clusters or structures that are spatially close to each other. And for some of these ligand and receptors, actually, it's known that they need to be close to each other. So you can kind of select for realistic ligand receptor interactions because you do have the spatial information available. Yeah. On the other hand, if you don't want to look at genes but just at clusters, there is of course you could look at the spatial distribution of clusters in tissue. So you could, for example, we provide permutation-based neighborhood enrichment scores that essentially tell you which clusters are more likely to be close to each other. So are more likely to be close to each other than would be provided by random chance. So this is also adjusted for the size of the clusters, of course. So here I'm showing a mouse brain gastrulation data set, I think. Sorry, a gastrulation data set. Yeah, so sorry, I get a bit confused. We have a lot of different data sources that we use with Squidpy, but here, yeah, essentially you can see that some of these clusters, I highlighted some of them on the right-hand side in the actual spatial image, and you can see that these clusters are ones that are kind of scored as being enriched for each other. So I'm not sure. Sorry, you can't see my cursor. This is the let me see if I can get the names. Well, this is essentially the red dots on this matrix on the right-hand side that you can see that I colored in the actual tissue image, and you can see how they're actually close to each other and kind of closer to each other than would be expected by random chance. So for this, you can figure out, well, which clusters, maybe which cell types are close to each other and are enriched with each other. In addition, we have something that is called the co-occurrence score. And this is essentially similar to the neighborhood enrichment. So it also looks at the relationship between two clusters, but it does this at different scales in the tissue. So this allows you to really see how clusters are related, because some clusters might actually occur at a certain distance, so always at a certain distance to each other, and then they co-occur much more at this particular distance, but not at other scales. So with this score, you will essentially get a graph like the one that is seen on the lower right hand side here, where essentially on the x-axis we're plotting the distance. So this could be in micrometers or something like this from your cluster of origin, and then on the y-axis we plot kind of the probability that a particular cluster co-occurs at this particular distance to the root cluster that you were looking at. And this can be quite helpful to characterize your tissue and to quantify differences between different tissues, for example. In addition, we also you can also calculate the Ripley statistics, which essentially tells you something about the degree of clustering or dispersion of a certain cluster or if a certain cell type. So this is not comparing two different clusters like the co-occurrence score or the neighborhood enrichment is doing, but this is really just looking at one individual cluster, one individual cell type. How is it distributed throughout the entire tissue? And this can give you an indication of whether there's kind of clumps of tissue that have the same, let's say, cell type, or if this cell type is distributed homogeneously across the tissue. This is something you can calculate with the Ripley statistic. Right. So for all of these tools that we have implemented to extract spatial patterns in the tissue, we have this lab called Spatial Domains. This is essentially shows you an example of how to use all of these functions that I've just described, and also gives you a bit more information on what the return type is and how you can plot the results and so on. Okay. Great. In case there is no questions for this particular part, I will then move on to the image part. So essentially, like I already motivated, in addition to dealing with the spatial graph, many of these spatial methods provide large microscopy images. And for this, we developed this image container, which essentially is just a wrapper of X-ray. And it's designed to be quite flexible and quite fast to use, because we use Dask to just lazily load these large images. If you've ever worked with histological images, you know that they can be quite large, and it's not really possible in some cases even to load them all into memory at the same time. And this is why this image container is at least designed to deal with this by just lazily loading whatever regions you're currently interested in. But actually for all of this, maybe before I go on, I want to mention that this is something that we're currently in the process of redesigning, and I will say more to this in the end. So I know that with the image container there are some issues and some things are not as seamless as they should be, and this is something we are currently in the process of standardizing, also with the rest of the community using images, and especially histological images. Right. So with the image container, essentially it's like any other image in Python, and we have implemented a couple of convenience functions that allow you to call some pre-processing functions on the images, like just converting it to grayscale, smoothing it, and things like this. The nice thing about these functions is that they can run in a parallelized fashion over the entire image. So again, it's quite fast if you have a really large image. If you have a smaller image, you wouldn't necessarily need to use the image container, because you could just use scikit image on its own. In addition, we provide a quite simple cell segmentation method, but also that's just based on thresholding and a watershed segmentation to kind of disentangle individual cells. But there's a lot of external tools that can be used. So we've kind of provided plugins for Statist, Telpos, and also Cell Profiler. And the nice thing that this image container does is, like I said, it can be parallelized. That is, it kind of iterates over the large image and just processes individual smaller crops. And this essentially allows you to also use custom deep learning methods. If you have a super nice deep learning based cell segmentation method, you could also use it with this image container. Yeah, and finally, we provide a module for extracting features from the images. And this is essentially a way of compressing or of making the information contained in the images similar to the transcriptomics or more generally the omics information that we have. So essentially to compress the image down to also a matrix that is observations times features. And observations in this case here, if we take, for example, if we just consider spatial transcriptomics, the observation would be an individual spot. So what we can do with this extract features function is we can iterate over all the spots, extract a crop, that is centered on this particular spot and extract some features from this image crop. The features could be something simple like just like a texture of the image. They can be just the intensity, just the gray value intensity of your image. It can also be a bit more complex, like counting the number of cells, like I already alluded to in the beginning, counting the number of cells in every spot and putting this as a feature in your observation matrix. In this way, essentially what this allows you to do is it allows you to extract more features, more information from the images that might not necessarily be contained in the genomics profile. And this can allow you to kind of combine image and omics information together in nice ways, or to correlate them, or to see in how far is morphology also contained in the transcriptome and all of this. So this is what this extract features is for, and what it would allow you to do. Yeah, and essentially this also provides then a close integration with undata, because the result of this extract features function is an undata object that contains, instead of gene information, it contains these features extracted from the spots. Yeah, in addition, we can also do interactive visualization, like I already said, for the image container. And yeah, oh yeah, sorry, I forgot I had some animations here. So these are some examples, but actually you will also see all of these things in, or at least some examples of the segmentation and also of the visualization or the plotting functions in this Spatial Transcriptomics image analysis lab that we are also going to look at this afternoon. So this will deal with essentially all of these steps: the preprocessing, the segmentation, and extracting features from images. Right. Okay. In terms of the interactive visualization, I just want to show you. This is essentially just a Napari plugin where we provide, on the right-hand side, we provide kind of the information that is contained in the undata. So the nice thing is you can call this with an image and an undata object, and then this allows you to overlay the information in the images, to overlay the information contained in the undata on the images. So for example, here we're showing how to overlay individual, like a clustering that you have in the undata, or individual gene expression levels on the image, and this in an interactive fashion. So this is, yeah, that's quite a nice interactive exploration tool. I think that is quite nice to use. Yeah. Okay. Great. Right. And maybe as a last point here, I want to mention that we also have more static plotting functionalities. This is in the most recent version of Squidpy. We've Giovanni has provided a lot of these spatial plotting tools that essentially extend and improve upon the spatial plotting function that we had before in ScanPy. We are now moving this all to Squidpy. This plotting function will still be left in ScanPy for backwards compatibility, but if you want to use the kind of the more advanced plotting that Squidpy can do, then you could use the spatial plotting functions that we have in Squidpy. And essentially what this allows you to do, it allows you to plot, first off, if you have several images in your spatial undata object. This can allows you to plot all of these images. It allows you to overlay segmentations, so not just plotting kind of round dots on the image like a scatter plot, but actually plot the segmentation mask that you may have stored somewhere. It also allows you to plot the actual spatial graph, and of course color it by clusters or gene expression levels or whatever you're interested in. There is a couple of tutorials on this on our website already, so you can also check this out to see what kind of functionality there is now. So as a very last point, like I already mentioned, currently there might be a couple of issues or with the image container, so it's not as flexible as we would like it to be, and this is why we're now really pushing towards moving towards this OME-NGFF a way of defining spatial data and storing it. So this is essentially a huge community effort to provide next generation file formats for storing bioimaging data. So this is what this OME-NGFF effort is for. And what we're now doing is we're proposing to actually use undata within this specification. And we are essentially imagining that we could store all of these different types of spatial images that you may have. So you may have, like I already said, you have these raster images, but you also have single molecule data. And there is different ways to store all of these different types of representations that you have in an efficient way. And there's also ways you could transfer or that you could transform your data. So you could go from a single molecule image where you essentially just need to store the X and Y coordinates of each molecule. So it's a very sparse representation, but you could go to a raster image. And because some of the cell segmentation methods might only work on raster images. So this is what we are envisioning for the future, to kind of have one nice container of all of this data, which is essentially what we thought the image container might be, but we realized it's much nicer to actually leverage the all the community efforts that are already being done in this direction of the OME-NGFF formats. So this is where we're kind of wanting to go to standardize the way of representing, especially the images. I think for the graph representation, everyone is agreed that the undata is a really nice way. It's just that the images that we're still struggling with, because it's not so nice to just store them in the undata. They can get very large, like I already said. Yeah, but if you are interested in this topic, there is quite a nice talk by Luca on this topic. Again, I will make these slides available to you, so you can also check this out. Yeah, for what is going to happen in the future with Squidpy. Right, we have quite extensive documentation on the Read the Docs with a lot of tutorials. Actually, there is quite a nice API. There's, like I said, tutorials and a lot of examples and so on. So if you have more doubts of what any of the functions are doing that we'll be covering in the labs, you can also go and check all of these tutorials and API examples out. And hopefully this already answers a lot of your questions. Yeah, finally, I just want to thank the kind of core development team of Squidpy, all of our other collaborators, and yeah, thanks for your attention. And please, if you have any questions, let me know.